Next up, we have oral presentation 11, Sanger, California, Mission 12 Flight Experiment.
Good afternoon. We are from Sanger Unified School District, located in Sanger, California, and today we are here to present the effects of microgravity on the developmental stage of brine shrimp. I am Jacqueline Ramirez, and I am a collaborator for this experiment.
Hi, my name is Austin Griesner, and I was a co-principal investigator for this project.
Hi, my name is Sean View, and I am also a co-principal investigator for this project.
Hello, my name is Lisa Rocha, and I'm a collaborator for this project.
Scientists and researchers have been discussing going to Mars for decades. A trip to Mars takes about 3 to 4 years, and learning to produce food sources in space will allow humans to live in Mars or in space for extended periods of time.
We believe our experiment could give insight to growing aquaponic systems for long space travel. Aquatic animals would be a good source of food. Brine shrimp can help us understand aquatic animals in space better.
For this experiment, we used a Type 3 FME mini lab separated into 3 volumes by 2 clamps. And in the first volume, we have 160 brine shrimps in cyst phase, and for this we used a tiny graph to approximate the number of eggs that we would be putting in. And in volume 2, we have 5 milliliters of distilled water mixed with a teaspoon of marine salt. And in the third volume, we have 0.5 milliliters of formalin.
We changed our experiment design a few times during the ground truth experiment, and we discovered that 5 milliliters of salt solution was the maximum amount we could fit in the mini lab and leave room for the other 2 volumes. Our original experiment had bleach to terminate the experiment, but we learned that formalin preserves better and also kills the brine shrimp.
Once the experiment is on the International Space Station on Crew Interaction Day, Arrival Day, they will unclamp Clamp A and shake gently for 5 seconds. No interactions will be done until 2 days before departure. On this day, they will unclamp Clamp B and shake gently for 5 seconds.
The reason we chose brine shrimp for this experiment is because they are in a dormant state while in their eggs. This egg is called a cyst. Once the eggs are in space, then clamp A will be taken off and the brine shrimp will be exposed to the salt water. They should hatch in 5 to 9 days. Once clamp B is taken off, the formalin that is in the third volume will kill the brine shrimp.
Throughout this experiment, we have 3 major benefits. We are investigating how brine shrimp can develop in microgravity with high levels of carbon dioxide and low levels of oxygen. Because of these harsh conditions, if the brine shrimp survive, we can study how they would also have successfully, if they survive, we can study how they would also function in a natural environment to have successful aquaponic systems for space travel in the future, as discussed before. Once that is attainable, scientists can use that information to use larger aquatic animals as a food source.
We would like to thank our sponsors, Walmart, California Space Grant, Subaru, Sanger Unified School District, Fairmont School Boosters, and Sanger High School. Thank you.
Do we have any questions?
Hello, my name is Felipe, and I'm from Fitchburg, Massachusetts. And my question to you was, how does this research apply to marine-dwelling organisms that maybe aren't crustaceans?
Can you clarify what you mean by that?
How could this process be, or this research suggest, I'm sorry, like, yeah, could your research show that, like, there's a correlation that this process could be used on other marine creatures besides brine shrimp, like using other forms of crustaceans or other organisms that aren't crustaceans?
Yes. This is used so that we can learn about brine shrimp, so that can be transferred to other marine species as well.
Okay, thank you.